By the 1960s, it was known that DNA directs the synthesis of RNA. It was also known that RNA is the template of protein synthesis. Nuremberg and Matthaei used this knowledge to gain an understanding of which of the three known base codons coded for which amino acid. They began by preparing extracts made from E. coli cells. These extracts contained all of the components that are required for protein synthesis, except for mRNA. They contain tRNA, ribosomes, amino acids, and amino-acyl tRNA synthase. However, they learned that without mRNA, protein synthesis did not occur. Next, Nuremberg and Matthaei mixed a poly-U RNA with the E. coli extract and radioactively labeled the amino acid, and a protein that consisted entirely of phenylalanine was produced. Using this mixture with the poly-U, they could only create a protein with phenylalanine, and the protein was radioactive, which meant that UUU coded for phenylalanine. Nuremberg and Matthaei repeated this process with a poly-A, poly-C, and poly-G. They found that a poly-A would create a protein made of lysine, poly-C would create a protein made of proline, and unfortunately, they were never able to get poly-G to create a protein. The results of these experiments proved that RNA was required for protein synthesis. It also led them to discover that the sequence of the RNA controlled which amino acids were in the protein. Later, Nuremberg teamed up with Leder and performed an experiment to determine the length of a codon. The experiment used a mixture that included an amino-acyl tRNA charged with a radioactive phenylalanine, a ribosome, and a poly-U RNA. Nuremberg and Leder knew that amino-acyl tRNA participated in protein synthesis and that they could attach to ribosomes in certain situations. Nuremberg and Leder created mixtures that had tRNA molecules with radioactive phenylalanine. One of these mixtures included a poly-U RNA while the other did not. The mixtures were poured over a cellulose nitrate filter. Cellulose nitrate filters catch only ribosomes. After the mixtures were poured over the filter, only the filter that included the poly-U RNA was still radioactive. Nuremberg and Leder learned that the poly-U RNA made the tRNA stick to the ribosome. This is why the mixture that did not include the poly-U RNA was no longer radioactive after being poured over the filter, as the tRNA was not stuck to the ribosome. Through this experiment, they discovered that 3 nucleotides was the minimum number of nucleotides that could be used and still cause reactivity. This means that there are 3 nucleotides in a codon. They repeated this experiment, charging the tRNAs with different amino acids to see which nucleotides they would bind to. This is how the genetic code was created.